So many neurons communicate with each other via chemical synapses. There are also electrical synapses, but chemical synapses are the typical type of transmission between neurons. So we're talking about signaling between neurons, or even neurons and their postsynaptic targets. And the two major players, two major participants, are the presynaptic neuron and the postsynaptic cell, whether it be another neuron or a gland or a muscle effector cell. And they communicate at the synapse, which is just this entire zone of communication between them. And this particular type of synapse is a chemical synapse. It's converting an electrical signal, which is action potential is propagating down the axon here. You have an action potential, and that's an electrical signal. It's facilitated by ion flux across the membrane, which changes the voltage. It's an electrical signal. And then it reaches the end of the presynaptic neuron, and at the chemical synapse, this electrical signal is converted into a chemical signal. And once it crosses the synaptic cleft and gets to the postsynaptic cell, it's converted into an electrical signal once again. So at the chemical synapse, you have to have this conversion of electrical to chemical back to electrical again. So let's get into the steps of chemical transmission. So as I said before, here we have a presynaptic terminal, and that action potential is coming down the axon. Here's the myelin sheath that usually ends a little bit of distance from the presynaptic terminal. And we have an action potential, so that's depolarization of the membrane coming down. So we can start with action potential. That arrives at the presynaptic terminal. Depolarization ensues, because that's what the action potential is. And that depolarization of this presynaptic terminal opens voltage-gated calcium channels. And these are in the presynaptic neuron. So when voltage-gated calcium channels open, calcium flows into the presynaptic terminal. Right, so we're just going to do this because it's flowing down its concentration gradient. So now there is calcium influx, and this calcium influx leads to, via a series of somewhat complicated reactions and interactions between a bunch of membrane proteins, it leads to synaptic vesicles that are already ready and contain a bunch of transmitter molecules. They're just kind of sitting here waiting to be released. The calcium influx mediates their fusion with the membrane and release of the transmitter. So that's a type of exocytosis, and it's called calcium-mediated exocytosis. Just a fancy way of saying that the calcium influx from the action potential, depolarization, causes the fusion of synaptic vesicles with the membrane, and this fusion results in the release of that transmitter into the synaptic cleft. So let's draw a little picture of what's happening. So the membrane of the synaptic vesicle fuses with the membrane of the presynaptic terminal, just releasing that transmitter into extracellular space. And now that that transmitter is released, much of it diffuses across the synaptic cleft and binds to postsynaptic receptors. So this calcium-mediated exocytosis is just a fancy way of saying this transmitter release. And this transmitter release, these transmitter molecules bind to postsynaptic receptors after they diffuse across the synaptic cleft. And this right here is the synaptic cleft. These are postsynaptic receptors. This is, of course, neurotransmitter molecules packed into these synaptic vesicles here. And once this transmitter binds to postsynaptic receptors, which are always integral membrane proteins found on the postsynaptic membrane, and they have transmitter binding sites, they carry out some sort of effector function. And that's very broad because there are a lot of different types of postsynaptic receptors, but typically they result in the gating of ion channels. And the gating of ion channels can lead to a change in membrane potential, like depolarization or hyperpolarization, which then creates an electrical signal, a synaptic potential, as it's called. That is a form of an electrical signal that can then propagate down through the postsynaptic cell. That is some sort of effector function, which is dependent on the postsynaptic receptor and not the transmitter. So often you hear that transmitters are excitatory or inhibitory, but that's not necessarily always the case. The excitatory or inhibitory nature is determined by the postsynaptic receptor that the transmitter binds to. The transmitter basically just activates postsynaptic receptors that it can bind to, and it can selectively bind to only certain receptors. But one transmitter molecule, one type of transmitter, let's say glutamate can bind to many different types of postsynaptic receptors. And those postsynaptic receptors determine the effector function, not glutamate itself doesn't determine the effector function. So certain transmitters can be excitatory or inhibitory depending on the postsynaptic receptors they bind to. And even the same type of postsynaptic receptor can cause inhibition or excitation, kind of depending on the context of the situation, like depending on the intracellular and extracellular ion concentrations and a whole bunch of other factors. But the biggest thing to know is that the excitatory or inhibitory nature is determined by the receptor and not the transmitter. The transmitter just says, all right, you're good to be activated, and you can excite or inhibit the cell the way you see fit. That's basically what this is saying, except glutamate generally acts on receptors that mediate excitation. So that's why people are lazy and say glutamate is excitatory, and most of the time they're right, so most of the time it's fine. All right, so effector function mediated by postsynaptic receptors that were activated by the transmitter. And this is typically ion channel gating. Certain postsynaptic receptors are ionotropic, which means they directly gate ion flux. So ionotropic receptors have the receptor, so they bind the transmitter, and they have an ion channel in the same macromolecule. So this is kind of what these are showing. So you have the ligand binding site, and then you have a channel pore that can be opened or closed and allows ion flux. So these are called receptor channels or ionotropic receptors. and those are the simplest kind of postsynaptic receptors. So transmitter binding causes the ion channel to open, thus mediating ion flux and converting the chemical signal, which is the transmitter, diffusing across the synaptic cleft, into an electrical signal. And it may be a little bit weird because the electrical signal is also mediated by the movement of chemicals, because ions are a type of chemical. But this electrical signal is facilitated by the local movement of ions. So if you think about an action potential, the ions that start the signal, that move over here, are only moving locally across their membrane, and then they cause kind of a domino effect of ions moving locally across their membrane. So the ions that started the signal are not the same ions that end the signal. The ions are not like moving all the way down the axon themselves. They are just basically moving locally across the membrane. The change in voltage is the electrical signal, and that's why it's considered electrical and not chemical, even though ions, the charge carriers, are chemicals themselves. A good analogy is people in a big audience or, I don't know, ball game or whatever, doing the wave. The signal there, everyone stays in their respective seats and they just raise their arms up when the wave comes by. So the signal that's moving is the wave, not the people themselves are only moving locally. So that is the chemical synapse in a nutshell. Some people like to organize it into the transmitting and the receptive steps, although those are not always clearly distinguished. But transmission is like the presynaptic neuron is releasing the transmitter, and then the reception is the postsynaptic cell receiving that transmitter and carrying out an effector function based on that transmitter binding. The reason that they're not always easily distinguished is sometimes the transmitter doesn't bind to the postsynaptic site, but it can bind to sites on the presynaptic cell and then modulate transmitter release later on. Or it could bind to a third cell that's nearby, and that third cell could influence either the postsynaptic or the presynaptic cells and modify synaptic efficacy. So this is just the basics. There's a lot of complexity that gets introduced when you start looking at exactly what these steps look like in physiological conditions. But there you have it. Hope this was helpful, and I'll see you later.